Hello, everybody. Welcome to our coffee break with TRANSFAC. And my name is Alexander Kell. And today we'll be talking about tissue-specific transcription factor and their binding sites and how to predict tissue-specific transcription factor target genes. Actually, we have many questions like this. So can you please give me a list of genes which are targets for my transcription factor? So give me all target genes for transcription factor. And this question is, of course, relevant, but it's a little bit under specified because transcription factors are regulating their genes in various conditions, especially they're very specific in different tissue types or cell types. And this question, just without the context, makes not much sense. Really, you have to know which conditions you are talking about. Can be tissue, we are going to talk today, or cell type, or can be either other kind of conditions which you may have in your experiment under a certain disease condition or under stimulation of the cells. In each of those conditions, you have a different target genes for the same transcription factor. So basically, we reformulate, I would say, this in different way. We would say, actually, if you have a list of genes, all right, and these genes are expressed in your tissue, so maybe a tissue-specific list of genes, and then your question could be, which transcription factors are regulating my genes? Because it's a very important question. If you answer it, you can really understand the mechanism, how those genes are regulated. You can maybe influence on those expression. You can, maybe manipulate with those transcription factor, create your special tissue of interest, those tissue reprogramming and things like that. And then it's very important to know which transcription factors are regulating my genes. And if you don't know this, what you can do, you can simply scan promoters of your genes for transcription factor motifs, but that's not enough. Let me show you on one example. So I have here an example of one entry in Gene Expression Omnibus, and that's very recent paper, or it was not even published yet. So this data is from very recent experiment where these authors, they try to, construct sort of a tissue, artificial tissue. So they play with the cell types, and they are interested in heart muscles, actually, cardio development. And that's clear now that cells alone, one type of cells, is not creating a full functional tissue. You have to add to these cells all kind of different types of cells. So basically, you have to construct the tissue. So basically, they develop cardiomyocytes. And then they were adding to these cardiomyocytes at different types of cells, actually macrophages, which help those cardiomyocytes to become real part of a heart muscle. So they really accelerate development of cardiomyocytes into the mature ones. And so basically what they did then, they made expression experiment. So they run it with RNA-seq, and they did this in many different conditions. And especially for us today is interesting these four conditions, these two conditions which are here. So basically these are cardiomyocytes with all with fibroblasts. And then they put these macrophages, so which create them to develop further, or here they without macrophages, so which is cardiomyocytes, but not really well developed. So we'll compare these guys to these guys, and then we'll understand how those development of cardiomyocytes proceed, and then which transcription factors are involved in that development. So to do that, we need actually the data, and to have this data, we have to download it from this
entries. So basically you put it in your file. So this is the Excel file. So I have it open it here already. So I will increase a little bit the font so you can see it more clearly. So what do we have? So this is a list of genes, and these are counts. So if you do a RNA-seq, you know these are counts. So how many reads you have for each gene in each conditions, and in each column you have the conditions. So we have these 3 columns for replicas of the cardiomyocytes with macrophages, and 3 replicas of them of the actually macrophages without cardiomyocytes. Well, these are cardiomyocytes, but without macrophages. So then what we do, we'll actually try to analyze these genes. And first we have to find which ones are upregulated. And then we will analyze promoters of those genes. And before doing that, let me actually briefly introduce you how to analyze promoters of the genes. So let me share you the screen. So I have to, yeah, right. So I guess you see my slides. So you see these nice introductory slides of me showing all these parts of the body, different tissues. It looks disgusting, right? But anyway, so these are tissues where different expression of genes is happening, which is driven by different expression of transcription factors. But not only, I will tell you more secrets how this expression is regulated not only just by one factor, which is expressed in the same tissue, but actually by combinations of factors. Anyway, so the different expression of different genes can be taken, for instance, from this Human Protein Atlas, which is a very nice effort for a big consortia. And we are using this data for our analysis as well. So what we're actually doing, so once you have a gene and you observe this gene is expressed in a particular condition, so there is a gene, there is exons, and here is the start of transcription. So you measure expression of that gene. So we know that gene is actually regulated by transcription factors, which are binding, first of all, in the promoter of that gene. So here is the promoter, but also there are enhancer areas a bit more distance from the start of transcription, where also transcription factors are binding. And so which means it's important to know where they are binding and which transcription factor they have their motifs to bind. So for that, we collected a lot of motifs in TRANSFAC database. So I'm just quickly repeating what you probably already know. So we're collecting them in the form of positional weight matrices. So we have more than 10,000 PVMs, positional weight matrices, collected in TRANSFAC, and they are built from known sites, so by some in vitro methods. So we're collecting them from literature, from databases. And once we have these motifs, these positional weight matrices in TRANSFAC, then once you have promoters of your genes, then you can scan this matrix through the promoter and predict where this score of the matrix is when it's actually finding this site in the particular place becomes above a given threshold. Which threshold? That's a big question, and that's actually something what many people don't pay too much attention, but it's really important because transcription factors, okay, they depending on the condition, they may bind to their target genes very loosely or very strongly, and depending where you put your cutoff. So basically, this is a score of the hit where you predict your transcription factor binding site, and the score can be maximally 1, that's a perfect fit, and then it could be 0 when there is no hit, but in random case there is like something around 0.75. And then if you have one matrix, for instance this E to F in this case, then by drawing these curves, actually saying how many sites, true sites, you will lose if you put a cutoff something like here, and how many false positives you will get if you put this cutoff. So, and then you have a balance between true positives and false positives, which defines your cutoff. Still, you know, in different conditions, this cutoff can, this binding affinity of that factor to their target sites, it can vary depending on the environment of other factors of the target itself, which means we actually don't know which cutoff to use. And our tool actually finds this cutoff automatically. And that's a very specific feature of the match algorithm, which actually able to find the cutoff. You don't need to give it beforehand. You just give a set of genes which are upregulated, for instance, in particular condition, and then the algorithm finds the cutoffs by itself. All right, so we are coming back to our case. So we have this gene expression data, and then what we are going to do, we just go back. We are going to our tool, which you can log in from our website. Here's geneXplain.com. So you simply log in here, and if you don't have a account yet in our system, just register here, where you can actually register by putting your license key if you have account, if you bought a license for TRANSFAC, or you have it already in your library, in your university, or if you are just a new registration and you don't know, or you don't have a license yet, you can register a free account in the platform and try many things in the platform without even a TRANSFAC license. So there is some possibility to do a lot of things in the platform without that. But once you have your account, so we go back to my login. So then you sign in, and then you have a variety of products to work with, and we'll touch today a little bit of different product. Mainly, actually, you can look in the database TRANSFAC and check for transcription factors, what they're doing, what is their expression. Many information about transcription factors are stored in TRANSFAC, but you can also go to the platform, which is a tool to analyze your data, to analyze your omics data, to analyze the gene lists, to analyze your promoters, to do many things. So once you're in a platform, so you see those different fields which you can dive in with your data. If you have RNA-seq data or proteomics data or all kind of data, you can apply different tools and different pipelines here. But again, what we are going to do today, we want to analyze RNA-seq data, right? So basically, I have here in my account, so where I can open my project. So you have much less projects if you just enter. So you have just one project. So, but you can create more projects. So which I have done a lot during my work. And here there are projects. So, and we are talking about hard development. So it's again, I will maybe increase a little bit the font so you can see it clearly. So let me create here basically a new folder, which would be actually cardiomyocytes, for instance. And I will load here this Excel file, which I have just showed to you. So I click here to upload from my computer, and I select here this Excel file. Actually, let me see, I guess this is the one. This is the file. So it recognizes which actually, all right, so it recognizes here we have the gene symbol, so we can start. So, and so we have here this file with all the counts. Okay, so what's next? So of course you would like to have upregulated genes. And we have all kind of tools to do that in the field of RNA-seq. For instance, you can go to detect differentially expressed genes, and here we have various tools to do that. For instance, you can do it with Lima, and to do that, yeah, let me close this. Let me maybe make it a little bit
smaller so you see the whole screen. All right. So what you do, you just drag and drop this table here. You say that's unnormalized counts, and then you have to specify your conditions. So this is my, experiment, and this is my control. And that's it. So then you run it, and it's actually working to analyze your genes. So it started in R script. It's actually using bioconductor tools, and not only it's in this case, it's actually using a tool which is which we have in the Galaxy package, so which we put it here. So it actually run different tools depending on the need. So it will take some time. That's why I will go actually to the results of that, which I of course did before, and that's the table, which is a result table of such differentially search for differentially expressed genes. So you see here the log-fold change, and if you sort by it, so you can see the number of genes here at the top, which are very, very much upregulated, and the p-values and adjusted p-values are significant. So you can filter this table by special filters here. I don't show now because it takes a lot of time of manipulating this. Actually, I have already selected this gene list. So we have here 45 genes which are actually upregulated in these cardiomyocytes, and which have a positive log-fold change, and which also have a significant p-value. All right, so we have these 45 genes, and they are we can say they are very much specific for cardiomyocytes. Okay, so what's next? So we go basically here back to our tools, and we want to analyze this gene list. So here we have a gene or protein list entry where you can say, I want to analyze regulatory regions of these genes. So I click here and identify enriched transcription factor binding sites in the promoters and using TRANSFAC. All right, so what next? What you see here is a pipeline which will do many things. So I will explain it while it's working. So you drag and drop here this gene list. It needs actually a control set. So which means it needs a set of genes which are different from your set. And in this case, it's using automatically the set of housekeeping genes. So, we are talking here on cell type specific genes. They are very specific for a particular cell type. So they expressed in that cell type under very specific conditions. And as a background are those genes which are expressed everywhere and always, so-called housekeeping genes. All right, so we have them here, but if you have any other control, you can use it here as well. For instance, from the same experiment, you can use a set of non-changed genes, which we are not using right now. One also important thing is which set of motifs we want to analyze. So actually, of course, we can predefine this set and say, I'm interested just in one transcription factor. But in this case, we are kind of agnostic. We don't know which factors are binding. And so that's why we can apply the whole set of motifs, which is, basically a collection of vertebrate motifs in TRANSFAC. So we can have a look at this collection. It's called actually a profile. So we have profiles here. And this profile is a vertebrate human P001, which contained 8,000 motifs. So I remember I told you we have 10,000 motifs in TRANSFAC for all kind of species and organisms, and especially for vertebrates. Of course, that's most studied one. We have 8,000 out of 10. It's the biggest collection of motifs which you can find for the positional vein matrices. And they all have the cutoffs, predefined cutoffs. But as I said, the cutoffs themselves are here just for initial analysis.
The actual cutoffs, actual thresholds, and parameters are specified by algorithm itself. So we have this collection, and we applied all. So you see, this is vertebrate human P001, which means these initial cutoffs are allowing to find the at least one hit per 1,000 nucleotides. And then there are some parameters here, and the very important one is which size of promoter we are analyzing. So, and actually here, as you can see, by default, we have a downstream from TSS 100 nucleotides and upstream 1 kb, which is a kind of a default. It's a pretty big promoter, I would say, which include already a first, kind of enhancer areas, because actually the proximal promoter is very small. It's near TSS, but the bigger region which we can analyze, we are analyzing here is 1 kb upstream. Actually, we can analyze even bigger, but you can understand the bigger region you analyze in one chunk, the more noise you have. So 1 kb is kind of optimum. And by the way, here we have Q&A section in the Zoom. Please don't hesitate, put your questions right away. I will always watch what's going on in the Q&A. And if something is not clear, just interrupt me with this question. Also put any other question, I will address them at the end. And we'll have enough time to answer all your questions. All right, so and also, okay, so now it's ready to start. So we're going back to my folder. So let's specify a new name, and then we simply run workflows. And what this workflow is doing, so you actually can see here at the bottom of the on my screen how it is executed. So the blue boxes here are different steps of the workflow, and it shows which step is now running and what is the percentage. So it's first actually, first it takes two sets of genes. We call them yes and no. So a set of our genes and a set of housekeeping genes. And then it searches for transcription factor binding sites, which are actually enriched. It's actually searching them by a very quick algorithm. Once it found them, it then take those motifs which are enriched and then start analyzing them more carefully in order to see actually more information where the binding sites for these motifs are located in the promoters and so forth. So it's doing its job, so let it work. But we will have a look at the result of that, which I already did before. So actually, this is the folder which is created by this algorithm, and you have here a number of files which are which this algorithm creates. Okay, so it's actually already my previous run, my current run is finishing. You see, it's opening automatically the results in different tabs, which you can go through by clicking here. You see all the matrices which were found significant. You see the first maps. You see, other information. But let's go for more in details in this folder. And first, what we want to have a look is the set of enriched motifs. So this is a table of 238 motifs. We can sort them by the enrichment score, which is actually an adjusted score of frequency of sites found in the promoter of my target genes compared to the housekeeping genes. And then it calculates also false discovery rate for this site enrichment. And also it also checks this not only site enrichment by sequence enrichment, which means actually how many promoters from my set, from 45 genes, how many genes are actually covered by this motif. So it may happen that not only few of them are covered. For instance, in this case, SRF motif is enriched in general, in total, but only half of the of my collection contain these SRF sites. what is interesting for us, okay, first, maybe we can simply look at these sites, but in order to look at them, we have to open them here. And this is actually, as I said, this is a more detailed view of on those sites. So, and then to look at them, we can here it's sorted by p-value. We can sort it by yes to no ratio. This is enrichment score, as I said, and these are the already motifs which are enriched. Let's have a look, for instance, for this MEF2 motif, which we will talk a little bit later today. This is very specific factor for actually heart muscles. By clicking here, we can actually see where exactly those sites are found in my promoters of those genes. So you see, this is a list of my genes and 45 genes, and these are the sort of, this each bar represent a promoter area which we analyze. So the red box here is the beginning of the gene. And then actually here the start of transcription, and here the first 100 nucleotide, here the first 1,000 nucleotides, and then you see each arrow here is a binding site, and you can even look at the more details. What was the cutoff? What was the score of the site? What was the motif itself? And you can even go more in details with that site. If we have time, we can look at the more details of that. But what is interesting for us, so far what we found are just motifs, and these motifs, they just show what is a potential of that promoter to bind various transcription factors. But as we know, transcription factors are enriched expressed in different tissues. So that's why what we can do here, we can actually take this list of transcription factors, which we have here, which are all factors which are associated with these motifs which were found enriched. And then we can, what we can do, we can annotate those factors with expression values from this expression atlas, from protein expression atlas. So it's how to do it. It's very easy. So we have here in analysis tools, we have various methods to work with our tables. For instance, we can annotate the table with various information. So we take our table, and then we have to put this annotation source from another source, from another table, basically. And here we have pre-prepared tables from Protein Atlas. For instance, here the expression of all genes is here. So we drag and drop it here. And here you see all kind of tissues where you can can put expression in different tissues. So what we need here, the expression in the heart muscle, right? So, and then we annotate that table with this heart muscle expression. And so you see here this heart muscle expression. And if you can sort by this expression value, there are actually many factors which are not expressed in heart muscle. So they have zeros here. So although the motifs is there, but the transcription factor which is associated with that motif, the one which is, which we have, is not actually expressed, which means this factor is out of scope. So, and we, if we sort it by, by those which have a high value of expression, you see now those factors which not only have significant enrichment of the motif itself, which we have here, right? But it's actually also have a high level of expression. And if you go down, you see here, for instance, you see factors like this one, which have a very high level of expression by itself, and the enrichment of the motif is also very high. And by actually combining these two values, the expression and enrichment, we can find those transcription factors which are really playing a role in these particular conditions. So it looks like, have to do a lot of steps. That's why we automatize this a bit. All these steps of, converting in different, annotating with different expression, analyzing enrichment, combining these various values. So we combine it in a tool which we called Match Suite, which is actually a very easy to operate tool. Actually, I have to close first my platform. Let me go home here. So I have to, yes, right. So go back to start page. So I have to, if we go to the Match Suite now, so we have this easy to work tool where you can start analysis and you can actually run your gene set. And here you simply upload your gene set. For instance, I uploaded this many different gene sets, and we'll show this in a moment. And then you click next, and you select your tissue of interest. You click next, and it doing all this, and it generates the report. But before actually showing this report of that very automatic tool, let me give you a different example, which actually opens a kind of a new perspective. In a sense, let me just show you some slides of that. So I go back to my Microsoft PowerPoint. All right. So let's have a look at the different type of transcription factors. These are interferon regulatory factors, IRFs. So if I again increase font, you can see them. So what you see here is a classification of transcription factors, which was built by Professor Wingetter some time ago. And he actually collected all known transcription factors of human and classified them. And there is a class which is a class of IRF factors. You see there are a number of them, like starting from 1 to 9. And they all bind to the same motif, more or less. So you see there are different factors, but the motif itself is very similar. A lot of A's and CC in between. All right. So which means if you actually just look for the motifs in your promoters, you cannot tell exactly which factor is actually binding to it. But it's very important because if you look at if you look at the in this family of factors, actually there are more like IRF10 as well, but it's only for chicken. So the DNA binding domain of that factors is very similar. So they bind to the same motif, as I said. But other domains, this is a protein, structure of those factors are quite different. And these domains, they do many things. So first of all, they actually regulate genes in different ways. They cooperate with different factors in different ways. And also they expressed in different conditions. All right. So, and if you align them, you see them that they're indeed different. And if you look actually an expression for expression of these different transcription factors in different condition, different tissue, you see that they are very much different. So for instance, you here see RF1, which is mainly expressed, which mainly, it's expressed in many places, but the top expression is in granulocytes. So basically, these are factors which play an important role in immune cell development, but very specific subtype, granulocytes, right? But, the RF2, for instance, also in granulocytes. But if you look at, for instance, RF8 or RF10, no, RF8, that's right, this one. It's actually have in granulocytes, it's not expressed at all, but in dendritic cells, it's expressed very much. So that means actually you have really just finding motifs is not enough. You have to know which transcription factors are expressed where. And that's why what I did actually, I used here a special tool which helps me to create those different sets of genes which are expressed in different conditions. So here in this Match Suite, we have, when you start analysis and go to the gene set analysis, here we have an option which is called construct tissue-specific gene list. So if you are interested in tissue-specific expression, of course, as we can do, as I did, you can do experiments and you measure your gene expression in your conditions with RNA-seq or with any other type of experiments, or you can do it virtually, sort of. You can look in our database here, and you can select a subset here of genes. For instance, I'm interested in dendritic cells, right? Dendritic cells. And it takes all the information from the database, and it shows it in two-dimensional space, where here you have an expression of the gene itself, but you see expression of the gene in a particular tissue doesn't mean that this gene is specific for this tissue or cell type. It's maybe expressed highly in that cell type, but maybe it's expressed everywhere, you know, with a high value. It's like a housekeeping gene. So that's why we have a second dimension here, which tells you what is specificity of that gene expression in general. So those ones which have each dot here is one gene. Those ones which have a high level here means they are expressed very specifically in one particular tissue. And then by choosing like this, you can select a set of genes which you are interested, which may have a specific enough and high level expression enough. And then you proceed with that set of genes to your analysis. Yes, so always click on green button, so it will proceed you to the next step. So it actually now taking all these 164 genes, and it goes here. It also specifies the studied tissue, and here you can specify your promoter range. You can do it like for this proximal promoter, very narrow, like to minus 500. And it's actually a default parameter in this match suite, which is always very often a good choice, because if you find enrichment in the proximal area, it's definitely motifs are playing a role in regulating expression of that gene. You can, of course, specify a bigger range up to 5 kb. So then you click next, and then your program is ready to start. You simply launch your analysis, and then you don't need to do anything by converting whatever what I did you in the beginning. So so you understand what is the complexity of the analysis. But in fact, our main trend and here is really show you tools easy to work. So you don't need to be a programmer or need to be a bioinformatician by yourself. You just need to understand how to click on different places and understand a little bit of biology. And then you will be able to analyze your data set. So not waiting for until your bioinformatic people will be ready with the analysis. You can do it yourself. All right, so here you see several sets running here. It will take some time. That's why actually what I will do, I will go to the pre-analyzed data. And here you see I analyze the dendritic cells and granulocyte cells in two different runs. Let's have a look on the results. So by clicking on view results, you actually see the full picture of the results. And first, what it does, it actually sorts for you the transcription factors. And it's exactly doing it like I said in the beginning, by combining two more or less two values. So actually I can open here a bit more detailed view. So I want to open it a little bit more. So it combines two things. First, it of course calculates the enrichment of the motif, right? So, and this is the enrichment value which we've seen in that table. So 2.18 means it's this motif, for instance, is enriched more than 2 times in the set of my genes compared to the background.
And also, you have expression value of that transcription factor in this selected tissue, and also specificity of that expression. So, for instance, let's have a look at this line, and which is for us, that's the reason to run it in general. So you see here IRF8, which was put here in the ranking and the second place, which means it makes a prediction that RF8 is actually playing a role here, a very important role in regulating my genes. Why? Because the motif, and in fact, interesting, the motif which is associated with these factors is called IRF4. It may be misleading, sort of, but that's the fact. So actually, independent of the factor itself, which we use to build the motif, like here IRF4 was used to build the motif itself is capable of predicting binding sites for the whole family of transcription factors of IRF family. And only by knowing that this IRF8 is expressed in these cell types and combining it with this IRF4 enrichment, and also knowing this expression, it identifies that this IRF8 is actually a most important factor in regulating my genes in these conditions. And also telling this RF8 is actually number 1 out of 62 other tissues. So which means expression of this RF8 in the dendritic cells is the highest for the RF8 compared to all other tissues. So it's actually number 1. So that's where you identify which factor is actually regulating my genes. By the way, this tool also shows, of course, the exact information about binding. So that's you see here in these 2 other windows. So here you are your genes, so your 160 genes, and then each line here shows actually which transcription factors, so you have to open it like this, which transcription factors found the binding sites in the promoter of that gene, so in the number of sites actually. And then here below you see also the map showing each of that gene. For instance, in this case, this is the gene which is a CLE-C10A. And then here you see each of this arrow is a binding site, predicted binding site for our transcription factors, and we split it in 2 parts. So the first part, which below here, is enriched factors by themselves. So, and we see here, for instance, IRF4, which we were talking about, right? So this motif was enriched by itself, by individual motif. And that's why here. The other part is here, which is called CMA, composite modules. It tells us which factors are actually binding to this promoter of that gene in combination with each other. And this is a important tool which tells you that the tissue-specific regulation is not only governed by individual factors, which are very specific for that tissue, but it's actually governed by combination of factors. So other factors which may be not really specific for that particular tissue, but in combination with other factors, the combination is become specific. And that's, we can see actually in the report. So the tool generates the report, which tells you many things. You can read this about this report when you use this tool. So it reports you the factors, reports you all the sites in your promoters. You can also see a heat map. This will come to that a bit later now. But what I wanted to show you, these combinations of factors which it identifies. So actually what it takes promoters of these specific genes, and then it applies quite interesting machine learning technique, which is called a genetic algorithm,
Some people call it even, call it as a first kind of artificial intelligence. So which start to build a model, we call it a model, a composite model of several motifs, which altogether are happening more frequent in the promoters of our target genes compared to the background promoters, to the housekeeping promoters. And it found this model, which includes, important motifs for various factors, which are not really so specific for that particular cell type, but together, as I said, this complex, this motif combination is specific for that cell type, and that's how we can actually recognize which factor might be playing a role in that specificity of that cell type, even though the factor itself may be not as specific as the ones which are really enriched by themselves. All right, so one important heat map I wanted to show here. This goes a little bit even more in details about specificity of factor binding. Let me move this like this. So what it clusters the gene ontology terms together, and then so inside the granulocyte expression, there are some genes which are actually belong to different categories. For instance, there are immune effector process genes, cell activation genes, and so forth. And then this tool actually analyzes not only the whole set of genes, but it analyzes each subset of the genes belonging to each geo categories and find those motifs. I can make it even bigger to show you a little bit, and find those motifs which are actually specific for a particular subset. And for instance, RF4, which we are talking about, is of course, as we found, specific for the whole set of genes expressed in granulocytes, but most specific for the subset of genes which are involved in exocytosis, in leukocyte activation, involved in immune response. So with doing that, you can actually see a bit more in depth, in details, what's going on with the regulation of your genes. All right, so that's why you can really analyze gene expression of your genes in a very particular specific conditions using different tools. And I showed to you this platform, which is a universal tool to apply all kind of tools and do your own pipeline, more or less. Or the tool which is prepared for you, which is called MatchSuite, which can help to to analyze your data very easily. Just click number of buttons and you are there, and your genes are predicted, your transcription factors are predicted, and you know which transcription factor are really regulating your genes. All right, so with that, I would like to finish my presentation. And I'm really open for your questions. Please don't hesitate to tell me what did you understand from this seminar today. I'm going back now to the platform, so to show you what actually this platform in principle can do for you. So you may have various questions about these tools, about the database itself. And by the way, if you're watching this in YouTube, please put like. Also subscribe to our channel. So, actually, as I said, our mission is to give easy to use tools to biologists. So you don't need to wait those, evil bioinformaticians when they are ready with the analysis. You can really do your analysis no coding way. So you don't need to be able to code, to program. So don't hesitate to use this tool to do that by yourself. And also, I'm waiting for your questions. All right, so I have here, actually, we have a number of questions which were sent to us in between. So as you know, we have our coffee breaks
going regularly. And if you don't have time to answer to ask your questions here, you can send us your question by email or by just filling the form. And there were a number of questions concerning the other organisms, not only human. And yes, the TRANSFAC database is not only for human and not only for vertebrates. Here, for instance, in the platform, we have already prepared genomes for several model organisms. So you see here mouse, rat, human, of course, but not only. We have nematode, fruit fly, so Drosophila, Arabidopsis, also yeast, different types of yeast. So these are prepared, also zebrafish actually, prepared genomes. But you can also use your own genome if you have a genome of interest. You can upload the genome here in the platform, and we have also show how to do this in some of our seminars. And you can apply motifs from TRANSFAC which are related to these different organisms. Of course, you may not know so much about the tissue specificity of those factors of working in different organisms, but this motif enrichment can be done well. And going back to TRANSFAC, for instance, you see here in the profiles, among the profiles, as I showed, you have here the vertebrate profiles, which I applied to all vertebrate species, of course, human, mouse, rat, but not only, all mammals, and even simple vertebrate organisms, those who have at least one vertebrate, where you can apply those motifs. And that's important to understand that, again, because the motif just shows the DNA binding specificity, and many factors in the evolution, even if they evolve quite significantly in terms of regulation, in terms of tissue specificity, the DNA binding structure is very similar to other organisms, and that's why for instance, all vertebrate motifs can be applied to analyze all kind of species. Also for plants, we have quite a good collection of plant matrices. Of course, plants is a bit more difficult issue because they evolve for billions of years, which means most of the profiles which we collected here, most of the matrices are actually coming from Arabidopsis, and you have to be careful to apply it to a very distant plant, which is very distant from Arabidopsis. But generally, if you don't have anything, it could be a good idea to apply it to generate your hypothesis. Okay, I have a question here in Q&A. In the specific tables slide shown representing the platform, for instance, in case of Hox, on the header site model were shown to okay, they were shown several motifs. So, okay, how this can be interpreted? Are two motifs linked to the same to that specific factor? Thank you very much, Kanika, for your question, and that's very important that you have seen that. Let me go back to that table. Actually, maybe I have opened it here already. Yes, that's one. Right. Right. So you see what was the question. So we have here the one transcription factor, which is HOX-A11. So one particular gene encoding one factor. But here we have two motifs which are linked to this factor. Actually, in this is very easy case. This is HOX-A1106 and HOX-A1108. So these are two matrices which were collected for the same factor, but from a different experiments. So in fact, they were maybe from different papers or done under different conditions for the same factor.
So in TRANSFAC database, we collecting those motifs just accumulatively. If they are done in different conditions, if they are done in different paper, we're still collecting it without removing the previous matrix. So with that, we actually quite agnostic. We don't know which matrix is better than another one, even it's maybe done in the newest time or published in the Nature compared to other metrics which published in not as not as well-known journal, but still we don't know which matrix will be working exactly in which conditions. That's why we apply all matrices when we analyze our promoters. And in this case, they both to the same factor. So they're coming from the different papers. More problematic could be cases like this when, that's basically the same case, a factor TFAP4, but so many matrices linked to this one particular factor. All right. That's also like the same factor, many matrices just done in different experiments. But this case may be more complicated. This is a factor TFATC2, a particular factor from the family NFAT. And then you see actually two matrices from this particular factor and two matrices more general of NFAT, general one, and even NFAT1, which is actually a different factor. And that's exactly the case which I described today. Then actually we build the matrix from one factor, but since the domain is very similar in all members of this family of transcription factors, we can associate all the family members to that matrix. So because this matrix is just recognizing the motif, and this factor is capable of binding to that motif, even the motif itself was built for a different factor but from the same family, right? So that's why many different matrices can be connected to one factor. And other way around, actually, one factor may be connected to different matrices. Do we have here these cases? And in this case, they are all different. But it may happen that the same factor can be connected to two different matrices. So in this case, we don't have this case, but this is possible to have. All right. So thanks a lot again for your question. So if you have more questions, please put in Q&A. I'll be happy to answer. In the meantime, I will show briefly the other tools which we have here. Of course, I have an account which have many different tools, so that depending on your license, you have just one TRANSFAC, or maybe you have the full license to our tools, which include also TransPass and human PSD, which means, okay, that's very important. So you have found your transcription factors, but what next? So if you want to understand which pathways these transcription factors actually belong to, and which factors are regulated through which cascade of signal transduction, you can go, you can have also information about pathways, signaling, and also about targets, drug targets, and drugs themselves, which you have in human PSD. A lot of information are stored in these other databases. So TRANSFAC is not the only database which we have. Also, you see there's a match suite, and here is a very important tool called Genome Enhancer, which I really love this tool because you don't need to do anything. You actually open this tool and it gives you sort of let me open just to demonstrate you. It has a big you know, like you open your project, you have Dropbox here, and you put here all kind of files, FASTQ files, your tables coming from from RNA-seq experiment, from proteomics experiment, coming from maybe epigenetics, ChIP-seq experiment, a toxic experiment. And then the tool will understand what to do with all this data by itself. You have to specify some very few steps just to tell what is experiment, what is a control. And then the tool will do, for you all the analysis automatically. So as I said, I love those kind of tools. Please try it. As for the classical database, as I said, you can look for the information about your transcription factors. So for instance, RF8, which we were analyzing today. So you see it from different species, and if you go to that factor, that gene in human, of course there are names and which gene ontology terms belong to. But what is very interesting is here to which diseases this factor, this gene, this protein is associated. What are the biomarkers of those diseases? This is expression, and you see this in dendritic cells. Here is a top. It also shows regulation of that gene of that factor by itself. So which other transcription factor are regulating my gene, regulating actually expression of this RF8 gene. It's a factor, but it's also a gene itself. It has information from many ChIP-seq experiments, from ENCODE database. It has other information about pathways where these factors involved, all kind of motifs, and that's exactly the case which I was looking when one transcription factor is actually associated with many motifs. And so they all collected from different experiments. So for instance, this one is from ChIP-seq, and this one from Celex, from in vitro experiment. And so some of the factors, some of the matrices like these ones are compiled from genomic sites, and they have this Q6, which means they have a high level of evidence. So if you click on that matrix, for instance, you can see all the details. So you see the matrix itself. You see which factors are actually connected to this matrix. So that's the case when matrix is connected to many different factors, because they all have the same DNA binding domain. And here you have those known sites, 36 transcription factor binding sites, which are collected from various papers. Here you see the references to those papers. So these are really, each of that site is one work of one PhD student to find this site in DNA. And here the whole collection of this huge information is in one place. And then, so our team has aligned those sites. You see this is alignment here, and build this positional weight matrix, which is now used to recognize binding sites for these factors in new promoters. All right. So any new questions? Okay, so far we don't have any questions. I guess you will, if you have a question, you can send us this by email, or you can put it also in the form. So let me go to our geneXplain Academy. So that's one which I mentioned, No Coding Bioinformatics Academy. So which is our mission really to tell you how to analyze your data without really need to learn how to code, how to program, to write scripts. So and here that's the what we have today. And here you have a schedule for next seminars. So actually you can have your TRANSFAC as a download version and do whatever you like on your computer. So if you don't even see what you are doing, we never see actually. And this will be next seminar. But what I want to tell you today, that we are actually announcing
promoter hunting course. So this is a really intensive course, which we have in the beginning of June for 4 days. Actually, it will be done online, so not like you showed here. So we have also those courses which we run offline. So when you have people really coming to in one place, but this course will be organized online, and you will learn many things, basically how to analyze your promoters, and not just lecture. You will really do it hands-on. So we have hands-on sessions where you really have your data, and you will really apply these tools by yourself. And we have this actually several days, and then which is important. Once you sign for this course, you will get TRANSFAC for free for 1 week of that course. Also, a couple of days when course is finished, you still have your access. So you can also try to analyze your own data using TRANSFAC. So just go ahead, book your seat. So it is this course is not free, so it costs something for you. But if you have already a TRANSFAC license, it's fully free for you. Just go to that page and write your code basically here, your code of your university or of your license, which you get from your library or from email, which you get from us, and then you're freely can be assigned to that course and come here, and we will be happy to train you and to show you our excellent tools. All right. Thank you very much for your attention, and see you next time at our coffee breaks. Goodbye.